have Dr. Vincent Young, MD and PhD, who works at the William Henry Pittsburgh Collegiate Professor in the Department of Internal Medicine, Division of Infectious Diseases, Department of Microbiology and Immunology at the University of Michigan at Ann Arbor. And with that, we'll let you take it away.
A lot of the fields that you study, you get to come across the same people time and time again. Dr. Armstrong talked about how people can hear about the microbiome everywhere. And this is just from today. I just typed in the word microbiome into Google and did a Google News search. And there's all sorts of stuff: skin, gut, what happens with obesity. You kind of think about what's going on. We're back to the gut. Oh, SARS-CoV-2, and something about the microbiome might be involved there. So it's in the news a lot. And it's when you go shopping, you hear about the microbiome. You can go and hear about how the microbiome of your baby can be protected by certain kinds of creams, and certain kinds of bugs are great for your complexion. You can actually feed your own microbiome, or you can add to your microbiome as well. So there's a lot about the microbiome. Well, is this something that's new? Let me give you a little bit of historical context here. Whoops, sorry. So if you go to where scientists like to go, what's called PubMed at the National Library of Medicine, and type in the word microbiome, you find out that there have been over 100,000 papers that have the word microbiome published. I wrote this kind of introduction to physicians. I am a physician myself, and a lot of people were asking me about, what is this microbiome? And this is 4 years ago. And at that time, there were 8,000 papers that were published around that time. And I go, wow, it's really taken off. Well, if you look last year, we've gone from about 8,000 to 24,000 papers published that have the word microbiome. And it's really taken off over these past decades. Now, I'm going to date myself. I know there's some people here who are in high school or in college. I graduated from high school right on that second dot there. There weren't a lot of papers published with the word microbiome when I was leaving high school. Well, things have definitely changed. So this was just published last month in this so-called Young Medicine magazine. And I was interviewed by a 7-year-old who wanted to write an article about the microbiome and how the microbiome can help protect our gut, and how eating well can protect your microbiome. So things have definitely changed. But one thing hasn't changed, and that's you can never forget your teachers. So this was my high school. I'm glad it's still there, at least according to the web page, it's still there. I graduated in, again, 1981 from this particular high school. And just before the pandemic, I was giving a talk back in Buffalo, and I stopped in, and these were my AP English teachers from that time. And I wanted to point out, and this is something that Dr. Armstrong did very well, you need to remember to use your language precisely when you're trying to explain things. It doesn't matter if it's science, doesn't matter if it's poetry, doesn't matter, whatever it is, using your language in a particular way, and it doesn't have to be the same as everyone else. So I'm going to tell you my definitions of microbiome and another word that I like to use, which is microbiota. So there is not a controversy. The more common usage of the word microbiome is exactly as Dr. Armstrong talked about. But I use the word microbiota when I'm referring to the group of microbes that's in a particular environment and location. And for me, I like to think about microbiome, and I'll tell you why in a couple slides, not just the total community of microbes, but the environment that they inhabit. And this comes from the fact that ome is one of the roots, but you can also say that it's biome. And when you think of a biome, that's all the kind of plants and animals and living things that are in a particular place, but also those things are not alive, the soil, the rocks, because those all interact with each other. Living things interact with non-living things in a given environment. And depending what you're studying, for me, it becomes clearer to use these words slightly differently. When I say microbiota, I'm referring to the microbes. When I'm talking microbiome, I'm talking about the entire environment. And in the case of the human microbiome, for example, in the gut, it's shaped and is composed of things that are made and utilized by the microbes and the microbes themselves. But our gut also contributes to this environment. We'll talk a little bit about it. But I'm a physician, so why did I get interested in the microbiome? Well, here's a kind of a case that we'd see. Here's an older gentleman who's well, it's kind of funny, I should change that because I'm now older than the older gentleman I refer to, so I should update this slide that I've been using for a number of years. Anyways, he's admitted to the hospital with pneumonia, and it's not COVID. We assume that he has a typical bacterial pneumonia. And when someone has a bacterial infection, we can give antibiotics to try to treat it. And the patient starts getting better from his breathing. But for some reason, 3 days after he's in the hospital on antibiotics, he starts complaining that his belly hurts. He's developing diarrhea and actually becomes very severely ill. Well, that's kind of strange. Why would he develop something that looks like another infection when you're already treating him for an infection? He has a lung infection, you treat that, and now he starts developing what might look like he has a gastroenteritis, a gut infection. Well, this is the organism that I studied. It's now called Clostridioides. It used to be known as Clostridium difficile. People who study actually using the 16S gene and the genomes that Dr. Armstrong talked about, we like to rename things as we learn more about it. Whether or not that's helpful to everyone, it's still C. difficile. So this is a bacteria that can live in people's guts. It can also be found in soil. It can also be what's known as a spore former. And this is a form, so it actually is anaerobic. Oxygen will kill it. So how does it survive when it's outside of the body? Well, it forms something that's called a spore. And these spores are inert, they're protected from oxygen, and they're even protected from alcohol hand sanitizers. So the interesting thing, in the hospital, if you're only using alcohol hand sanitizers, all you're doing is helping spread C. difficile around. You have to use soap and water, so you wash it down the drain. The other thing, it produces a toxin, and this is what causes the problems there. It's not normally in healthy people, but as I said, it can be in the hospital environment. And I'll explain how when you're in the hospital and on antibiotics, how that can predispose you to infection with this particular bacteria. So this is a change on how we view infectious diseases. When we think of infectious diseases, we normally think of, okay, there's the host trying to defend itself. Here are the pathogens, SARS-CoV-2, C. difficile, whatever. These are virulent and somehow they can cause disease. But what we're understanding now is that the microbiota that lives in and on us can actually influence not just the host, but also pathogens. Well, again, how do we study this? We already heard about this. We're often looking at who is there, but we also want to know what they're doing, right? Because if we're going to understand exactly how they might be protecting us, we need to know who is there, but we also need to know what functions they might be carrying out to help us. And what are these functions? Well, you can break it down into several large groups. Well, one thing they can do is they can eat, right? And they can break things down. They can take certain kinds of starches that maybe you or I can't eat, but they can bacteria can break them down. They can also change drugs and other things by breaking them down. Okay, that's that sort of catabolism, bioconversion. Well, the microbes in our gut and on our skin and everywhere can also synthesize things. They're metabolically active. They can make vitamins for us. They can make all sorts of things that interact with us. And they can directly interact with our immune system, as we just heard about in the previous talk. Well, what does this have to do with C. difficile? Well, what we think happens is that when people have a normal microbiota in their gut, it's a stable community. This is going back to the idea of a biome and ecology, macroecology. It's a stable environment. And stable environments, it's difficult for other organisms to basically invade that environment. But what happens when we treat a patient with antibiotics? Well, if we're trying to treat a pneumonia, we can kill the bacteria that's causing the lung infection, but we can also inadvertently alter the normal microbes and change both who is there and therefore what they're doing. And this causes it so that this is a more invadable community. So when you get exposed to the C. diff spores, the C. diff spores can start growing, starts producing that toxin I was telling you about, and that causes the disease by damage to your intestines. Well, you might be in trouble because you have a bacteria in your gut. Well, we already had learned that we can use, over there. We can treat microbes with more bacteria, and we can give, with more antibiotics. We can give specific antibiotics that target C. difficile, and hopefully everybody gets better. The microbiota goes back to normal. But with C. difficile infection, about a fifth of the patients who were successfully treated, when you stop all the antibiotics treating C. difficile, the antibiotics that got them in trouble in the first place, they can recur. And they can go through this recurrence again and again. And we think that's because their microbiota remains in the susceptible state, never goes back to normal. So people got the clever idea, well, can't we restore the microbiota function somehow? Can't we kind of fix this recurrent cycle? And this brings up the thing that people always are kind of grossed out about and sort of fascinated with, fecal transplantation. Well, where can we give good microbes? They're in feces. This has been known since antiquity. Fermented milk products, fecal transplants. Chinese medicine talks about giving something that they call yellow soup, where they take feces, mix it up with water, let big chunks settle out, and take the liquid on top that's yellow. And that has microbes in it. They may not have known that, but they can actually treat a variety of illnesses with that. Okay, this is ancient history. Are we doing that now? Well, we can use that. I'm talking about these patients with recurrent C. difficile. A paper was published not even a decade ago where they did a placebo-controlled trial of giving feces to try to treat recurrent C. difficile. And it worked fabulously. You had a 90% cure rate if you gave feces, as opposed to giving yet another round of an antibiotic to treat recurrent C. difficile. We thought this was kind of interesting. Well, that was a funny story. My younger son started at the University of in 2018. And on the first day of class, he texted me a selfie. I'm going, what are you doing? And then I realized he was sitting next to a friend of his from high school. His father is a gastroenterologist here at the University of Michigan Medical Center. And I'm going like, well, why is he clowning around in class? Why are the two of them sitting together? Well, he then texted me this. On his first day of college, in his first biology class, they were talking about C. difficile and fecal transplants. The interesting thing about the kid who was sitting next to him is that his father, I know very well because he started the fecal transplant program here at the University of Michigan. So I knew the professor and I said, oh, if you had two guys who were clowning in the back and laughing about fecal transplants, it's because it was my son and his friend who runs the fecal transplant program. I'm not going to go into much detail about this, but we can study how the microbiome, in this case writ large, the bacteria, the things that they produce, the things that we produce in patients who are treated with fecal transplantation for recurrent C. difficile. This is a former postdoc of mine. She's now at Clemson. She studied how the microbiome can get helped by fecal transplant. And she also developed a mouse model because she wanted to be able to say, okay, it's an interesting thing to study it in people. Can we also do experiments in animals that we can't do with people? And she developed a mouse model of this as well. So where do we go? Again, this is just an overview of the microbiome. You've heard about association between the microbiome, multiple diseases, multiple conditions of health, and we're beginning to understand how do changes in the indigenous microbiota, how changes in those microbes can cause disease. And as we talked about, we're beginning to understand how we might be able to intentionally manipulate the microbiota to improve health, either keep us healthy or treat disease. And this goes into this idea, people may have heard about precision health. And a lot of this came about the fact that we were sequencing the human genome. And if we understood the human genome, we could understand how people are going to basically be genetically susceptible to diseases, how they might respond to diseases, and importantly, might have adverse reactions to treatments, including drugs, surgery, and other things. Maybe what we can add to that is if we can also study the microbiota, can we combine both understanding the microbes that live in us as well as our genes to come up with truly customized therapies to treat a variety of illnesses? I'd like to thank the people in my lab. I have been always helped by the people who are doing the work all the time, the people that I collaborated, funding, and I'd be happy now to take any questions. I can be reached also. I do like to tweet, and my Facebook is for my friends. Twitter is where I like to do my academic kind of social media. So you can kind of reach me here at A2Binney and happy to answer any questions.
Wonderful. Thank you so much, Dr. Young, for that exciting talk about the gut microbiome. Please, nobody try fecal transplants at home. Save that for doctors.
Yeah, that's a dangerous thing, because I won't send people to the Google, but if you look up home fecal transplant, as an infectious disease doctor, I would not at all recommend it, but it's out there. So I'll second that. Don't try it at home.
So now we'll move into the Q&A because we got a lot of questions. So first, but could you discuss superbugs in the relationship to the microbiome?
Studying C. difficile, we're studying how something it's naturally resistant to antibiotics is C. difficile. And that's why in this presence of antibiotics, it can come in. And also, on a perfectly serious note, that's why you shouldn't be trying fecal transplants at home. We're supposed to screen for many of these so-called superbugs, but occasionally they can slip by. And there are reports of people who have gotten and unfortunately died from multi-drug resistant organisms that were given via fecal transplant. So, yep, I think that's definitely something to think about.
Awesome, thank you so much. So the next question is, can your microbiome enter your bloodstream?
You bite into a carrot. We heard about the oral microbiome. There's a lot of bugs in there. You break down, you kind of have a tiny little break open a capillary. These bugs are pretty small. They can get in. You have a bowel movement. Bacteria will get into your bloodstream. But luckily, we have that immune system that we heard about so beautifully. So even though the microbes can get in, most of the time, if you're a healthy person with a healthy immune system, you take care of those small amounts of microbes that get in. Of course, that system can get overwhelmed. You know, we hear about constantly aspirating a little bit of saliva. Your body clears that out of the lung, or keeps it at a low level. But if something bad goes on, that's how you get a pneumonia. Most of the time it has to get in when you have a microaspiration and it grows there. So it can enter the bloodstream, but it's not something to worry about. Okay, so.
Awesome, thank you so much. So how can you tell the difference between a good microbiome bacteria and versus a bad one, and detect any mutations?
We're not great at that. That's what the hope is. That's what I was talking about when I presented that thing about how we can use understanding our microbiome to predict health. We're not quite there yet. We don't know enough yet. There are certain trends that we can see when we look at large populations, but the difficult thing is something that holds for 80, even 90% of a population means it doesn't hold for 10%. And so depending on what you're doing, we're not good enough to say, well, we need to do this really drastic treatment because you have a 1 in 10 chance of benefiting from it, or even a 9 in 10 chance. Depending on how bad the side effects could be, we might not want to do some serious things. If we find out that for 9 out of 10 people, cutting back on the amount of soda that you drink is good for your oral microbiota, yeah, that's okay, unless you're particularly addicted to Mountain Dew or something like that, in which case I would say go to coffee. But we're trying to get to that point where we can use that as a test. There are companies out there that will do tests, and my friends who are also physicians will come to me with some of these reports. My patient spent a few $100s to get this microbiome scan. How do I use it? I said, I wish I could tell you, because I can't. We don't know enough yet to be able to use it like some of the tests that we do when we draw blood or other things like that.
But you touched upon this, Dr. Young, in your previous answer, but could you tell us a little bit more about like how the food we intake and what types of food impact our gut microbiota?
Yeah, we like to joke sometimes, anything you do will change how the gut microbiota looks when you use a certain technique. But that's where it also is important. We talked about knowing who is there and also what they're doing. Because you can have 2 completely different looking collections of gut microbes, but they carry out the same functions for their specific host. So let's just go back to how food can change things. Well, one, I told you that there are certain kinds of starches that we eat, so-called resistant starch. We don't have the ability to break down these complex carbohydrates, but the microbes do. And in that way, they sort of even feed us. They can break it down into things that we can then absorb, and therefore they help with our digestion. Similarly, if we're eating those types of things, well, we can foster the growth of those in our gut. And I'll just give you one anecdote. This is a paper that I thought was really cool. People, a bunch of scientists in Japan were noting that there were some people who carried a bacteria that could break down a starch that was present in certain kinds of seaweed. And they also found out that that bug happens to live on seaweed. So it's kind of two things. They're bringing along the microbes that can bring it down. And if they eat enough seaweed, i.e. there are a lot of Japanese foods other than just sushi that has seaweed in it, well, then they're maintaining that particular bacteria in their gut. And when they move somewhere else, there are other studies when people have a microbiota that's prevalent in one area of the world, when they move to a new area of the world and adopt the food that's eaten in the new area of that new area of the world, their microbiota looks more like the people who live in that area eating that kind of food. So we can shape the microbiota, but also depending on what we eat, we can shape the microbiota, and that may also then change us as well. So it works both ways. So don't just think, if there's anything you take away, this is a very complex symbiosis. We're living together, okay? We're feeding them, they're feeding us, we're helping them, they're protecting us sometimes. So most of the time, the interaction is beneficial. Occasionally it breaks down, as we heard today, but hopefully most of the time, it's in a good kind of balance.
Yeah, life is all about balance.
Yes.
So are there any microbes that you can detect in the microbiota that specifically will give you indication that this person may develop a digestive tract cancer or Alzheimer's or any other type of diseases?
Yes and no. I mean, the direct one is one that Marty Blaser used to study a long time ago. H. pylori is one bacteria that if you get it at a certain time, you can get stomach cancer. That's a direct effect. The more complex ones related to like colon cancer and other cancers, there are suggestions there. We don't have the precise mechanism, and importantly, at least right yet, we don't have ways to try to alter it, to try to protect people from colon cancer yet. So there is data that suggests, and Dr. Armstrong will also tell us that some of the ones that are associated with colon cancer actually seem to normally be up in the mouth a lot more. Anything to add based on that?
No, I think that's my takeaway too. There's some things, yeah, that suggest a potential predisposition. Some things that might suggest you are more likely to develop a condition like, rheumatoid arthritis, some inflammatory condition, but the microbes aren't the only thing acting there. Your body is kind of taking what it's given. So the microbes are just one part of this complex system of disease. So there's no for sure when we're thinking about microbes.
And that's where I brought up with that precision health. Let's know what we can do. Let's know what the microbes should do. And then how do they interact? And then maybe we can get a better prediction. Because we have okay predictions from just human genetics. We have okay predictions just from the microbiota, but we seem to be missing things. Maybe we can fill in some of those gaps by looking at those interactions that we've been talking about all this time.
That's great. And that's honestly, students, something you can work on in the future if it interests you so much. We'd always love science advancements. So could you explain a little more about the microbiomes and relation to virus infections like COVID-19?
I was waiting when the pandemic started. I remember telling one of my friends, I can't wait till I get the first paper to review about the microbiome in COVID-19. And there are some suggestions that maybe it's a little bit worse in certain kinds. Again, these are mostly associations. And here's an important thing that I didn't say. Association means you observe A and B together. But association doesn't necessarily mean causation. Does A lead to B? Does B lead to A? Or are both related to a C that we know nothing about? So just because you see these things in association, and this, COVID-19 is a relatively new disease, and what role the microbiome may play, it's still up there. There are suggestions, as would normally be with all infections, as we already talked about with C. difficile, we talked about these superbugs. It can probably alter the host response and other things, so that it'd be unusual if there wasn't an interaction. But how important that interaction is, we don't know yet.
To talk about a few things we can look forward to in the future in terms of microbiome research and curing obesity or diabetes, or any other type of health.
I never thought I'd be working on this thing that I'd never heard about when I was in college, and even in med school, really. I'd heard about C. difficile, and people would say, oh, it's because the antibiotics kind of destroy the normal microbes and eliminate the ability to protect us. And I asked, well, wait, how do they protect us? How do we study them? And my teachers go, yeah, don't stop bothering me, kid. Because we didn't know. We had no idea. We didn't have these technologies to be able to study them, to be able to understand what was going on. So that's the fun thing about science. You never stop learning. I've been doing this for a long time. There's always cool new things to think about. Think about old questions with new tools. Think about new questions using old tools. All sorts of stuff can go on.
Awesome. So do you guys think that there will be any cure that could get rid of all bad microbes?
No, actually. How's that? Because exactly what Dr. Armstrong said, context does matter. And one bacteria that in one situation is beneficial can be harmful in the other. So there's no way to kind of just eliminate all microbes that could potentially harm us, because the context matters. And since we need them, we just can't sterilize ourselves. Living completely sterile, other than the fact that it's highly impractical, actually doesn't have good health effects. So.
Yes, you can buy completely sterile mice though for lab research.
You can buy germ-free mice, and they are kind of messed up. They don't have a good immune system. They actually have to eat more, right? Because they don't have microbes to help them digest. They kind of have other conditions that they develop over time. They functionally develop okay, but they get much better when you give them microbes. You can kind of reverse some of the weirdness that you see in a germ-free mouse by giving them some bacteria back, and viruses and fungi and other things. Let's not neglect the other small life that lives in and on us, but we focus a lot on the bacteria.
Awesome. So this is going to be our last question, and we have it a lot, and I'm going to combine a few into one. So this is for Dr. Young. Have you actually performed a fecal transplant? And then how do you actually transfer the healthy microbiome into somebody else, and what are the risks involved?
Well, we've talked about the risks already, and we do fecal transplants here at Michigan. You can, like I said, the simplest way they used to do it is you take feces and you kind of put it in a blender and mix it up, and you can use what's called a colonoscope that they use to look in a person's colon. You put it through the rectum and you can kind of go up, and they can then inject it as they're pulling the colonoscope out. You can simply give an enema. So you just put a short tube a little ways up and you just kind of drip it in that way. You can also have capsule form. There are special capsules that are meant not to dissolve until they go a little bit further down in the gut. You have to take about 30 of them, and you can treat them that way. Now, there are companies saying like, okay, feces is one thing, and we didn't talk about this, and this is probably the future. People are trying to, one, purify feces a little bit more so that it's just the bacteria, and particularly mostly the beneficial bacteria, getting rid of potential harmful ones. There's going to be a paper coming out in a few days talking about a company that passed a phase 3 trial. People aren't supposed to know about this for another 24 hours, so don't tell the news news yet. I didn't mention the drug, I didn't mention the disease or anything, but it's going to come out. I didn't mention the journal, it's going to come out. But there are other companies that are trying to start, rather than starting with feces directly, they're starting with feces, but then they're growing out all the individual bacteria, studying them in detail. And can we learn enough that we can build up a community that will be beneficial? And it may not be the same, well, actually, we know it won't be the same community to treat every patient with the same disease, and certainly not the same community to treat every single disease. So that's that idea of precision medicine, custom medicine. And we're trying to get there. So stay tuned, and probably you'll see more and more of these kinds of therapies based on studying the microbiome in the coming years.
Yeah, that's wonderful. Thank you so much.